So there's a couple advantages that Ion AmpliSeq gives us. I would say the first advantage is that it allows us to harness the next generation technology, but in a targeted fashion.
CE was the basis for the way we studied genes, and clearly this imposed upon us quite strong limitations in terms of the throughput, in terms of the number of genes we could look at.
You can describe the sample according to the subpopulations, molecular subpopulations that are inside. And this is impossible to do with the current methods.
So that's why we need definitely to have a panel that allows us to identify all the mutations, all the molecular alterations that are present in a specific tumor, so that we can really see which is the molecular landscape of each individual tumor.
The big advantage is that you're able to detect changes in a lot of genes at once, not just the particular ones that you happen to be looking for.
For every single PCR reaction, we would need something like 10 nanograms of DNA. And now we can do it in one reaction.
Nowadays we can go faster and we can study many genes at the same time. And very basically, or very simply, this is the main advantage of this type of technology for us.
The nice part of the panel, actually the AmpliSeq technology, is that you can get results on 20, 30, 40 genes by using 10 nanograms of DNA from formalin-fixed paraffin-embedded tissue.
We cover all the exons, at least 10 bases within the introns. So far we haven't missed any mutations with this panel. We've tested so far 65 different mutations, also in very difficult regions of the genes. So homopolymer stretches have been tackled.
AmpliSeq extends by a couple logs the dynamic range that we can quantitate gene expression.
The thing that got us excited from the very first glimpse of the data is that it really looks to be the most accurate representation that we've seen so far of allelic expression.
With Ion AmpliSeq, we can control which genes go into the set that we sequence, and so we can drive up the read coverage of those genes.
You have the machine and you have also the software associated with the machine. And for me it is important to have a whole solution in terms of genome sequencing from the tumor to the results.